Hello, everyone. My name is Yiyi. Nice to meet you. Today, I'm here to talk about my poster for ISMB and ECCB, CellDEEP. This is my first project during my PhD and takes a lot of time, so I'm excited to introduce it here. Now let's talk about the introduction first, which is why we do this project. Well, single cell RNA sequencing has been more and more important recently, not actually recently for a long time. So the differential expression is a very important step in it, but there are some questions. So it's because single cell data is so noisy, it has a lot of zero. It might be biological zero, which means the gene just doesn't express, or it can be the technical zero, which is dropout. That means for technical reason, they didn't catch it. Anyway, we will have a lot of zero. So that leads to the differential expression not as trustful as scRNAseq did. So I will talk about mainly two groups here, but there are more than two. It's just our project focuses on the gap between these two. So the single cell RNA-seq-based method is the first step, which is the method we use in serine pipeline like MAST or Wilcox, and also there are some DSEQ too. The advantage of this kind of method is it considers the single cell level information, which is the whole reason that you want to single cell for. Well, the not good part is it has a lot of possibility. So because because the data is so noisy, it can't capture all the signal, or it will catch a lot of false positive signal. Another type is pseudo-bulk-based method. So they are the method originally developed for bulk RNA-seq, like DSEQ2 or HR or Lima. And what it did is merging all the cells together and to make it like bulk RNA-seq. And by doing that, it will decrease the false positive and make the result very robust. Well, the bad point, as you probably can imagine, it lost a single cell level resolution. So for some genes, maybe very lowly expressed, or maybe it's just in a small population of the cells that are expressed, it probably will be ignored. So we are aiming on a gap between them two, trying to find some way to solve the questions. And we named this method CellDEEP. And here is the introduction of CellDEEP. So as you can see, single cell RNA-seq-based method is focusing on the single cells, and pseudobulk is merging them all. So what we think is, why can't we do something in the middle, like pulling some cells, but not all of them? That's CellDEEP. And to do that, we designed two selection methods. Number one is random selection, which is just randomly pick something. You can see it here. And the next one is k-means selection. So that is select the cells more like each other. The in our original design, so that means the k-means selection will select the cells more like each other, and random will be more dislike. It's just a two-way to do it. And we also did another two ways for the how do you aggregate cells. So after you select cells, you need to pull it, right? Then you can get the meta cells we're looking for. So when you pull it, how do you deal with read counts? We design the sum or mean. So sum just sum them all together, and mean is mean expression. That's the two-way we get. Now let's talk about the result. So the first result would be the result of simulation. So we simulated 18 datasets from the two pipeline. They are all published, and I have the reference lower in the poster. You can see it in PDF. The first is Muscas. The second is Zimmerman. What you can see is, and let's go through from A to B so that we can go. So the first thing I look at is which strategy of CellDEEP is better. And look at the plot A. We have precision and sensitivity. We actually have four, including false positive and accuracy, but because precision is a reflect of false positive, and sensitivity can not really seem, but they can actually reflect each other. So let's just look at them. You can see in all this strategy, oh, right, and for CellDEEP, besides K, which is k-mean selection, or R is random selection, mean is mean read counts, sum is sum read counts, like I introduced. I also have a differences between after you do it, do you run MAST or do you run DSEQ2? Technically, we can run all the D methods you like, but I just picked them two here as they are probably the most popular methods. Now, if you can see the precision, it's basically and the sensitivity. In Muscat, it seems like this olive yellow. That's random sum D6 is the best. And if you look at Zimmerman, that I will see the two simulation framework give quite different results, but you can see the best method is still this one. So random sum D6 is the best. So you keep going with it. Um, then another thing I did is I'm not sure how many cells to pull. I didn't show the result here, but I can tell you in simulation, when you pull more cells, the result is better. So that's one way choose the 20 cells polling method here. So then after we do it, basically we make sure random selection, sum read counts, and D6 is the best combination with more cells, which is 20 cells. Then I want to compare it with single cell and seed block, just see that is it doing what I wanted to do. So you can see here in the precision, CellDEEP is in the similar level with seed-block in Muscat. In Zimmerman, it's not similar, but you still can see there is increasing. So it's between seed-block and single cell. That's what I want. I'm happy about it. And for sensitivity, uh, this one is hard to see, but still you can see CellDEEP is in the good group, like it's better than not better than all of them, but it's still in the top rank. And if you look at Zimmerman, yes, it is the highest one generally. So in simulation, we are happy. We're happy about the performance. Uh, it keep the phosphative, which is also precision, at similar level as seedbulk, but even higher sensitivity. So happy. Then, but the next thing is I need to test it on real data, right? Simulation can't explain everything. So that's why, so let's go to result part 2, which is a real dataset result. Well, in real dataset, the issue is we don't know the ground truth. I can't just simply test the precision and phosphative like I did in simulation. So I designed two ways to do it. Number one is a p-value non-hypothesis test. What I did is basically I select cells from same group. That is to see they are both from healthy or they are both from patient, so that there shouldn't be many D genes between them. Technically, they should be the same, but maybe there are a little bit, but shouldn't be too much. And after running the D method, we make a histogram of the p-value. And for the p-value less than 0.05, they are all phosphative because there shouldn't be any real differences between these two groups, right? So if you look at this figure 1, single cell mass is terrible. It has a lot of phosphative, which is what we expect. Single cell D6 is good, actually. Not so bad, but still it has a little bit. And seedbulk, D6 and Lima, they control it very well. Like the p-value equally distributed between 0 and 1. That's what it should be. Now how about CellDEEP? Here I pick the top 2 strategy and still test all the strategy because I'm not sure the best strategy in simulation would be also the best one in real. So I test all and then I select the top 2 here. The top 2 is random in mass and random in D6 So you can see it's controlled very well, like seedbulk. I can tell you the number is not really seedbulk, but you can see like for mass it decreases a lot. For D6 it also decreases. So happy, it still controls phosphative like simulation. It's just the strategy, not that same, but let's leave it there and keep going. The second way is the geopathology test. So because we are using the data from the COVID PBMC public dataset, so we know between the patient and the healthy people, there are some biological activity you should catch. Like after you get the DE, you run it with geoanalysis, you should have a list of biological activity, right? And we can pick the terms that should be there. For example, for the virus-related activity, it should be there. And for the inform response activity, yes, you should be there. So our Dom, which is my second advisor, and Domenico Soma here, he picked about 90 Go terms that should be there. So then what we did is simple. I just run the D, run the geo, check how many D genes you get and how many geo pathway you get. So you would like as more as geo pathway you can find, but not too much D gene. Because for example, if you look at the pseudobok lemma here, this figure B is a CD8 cell type, CD8 cell cluster. You can see it catch about 1000 gene, but only 15 Go terms from the 90. And for the CellDEEP D6 it has 116, but got 17 Go terms. So apparently CellDEEP D6 is performed better than CellDEEP Lima here. Lima get a lot of positive. So that leads to another parameter I need to check, that is the true positivity ratio. It's simply divide the Go term, like 43, divide 408. I get a ratio, just to roughly say how many positive you probably get. So you want this ratio as high as possible. So combine these three things. I can see in CD8 the best performance strategy is a random mean D6 So then it goes to, okay, you are in this cell type, right? How about other cell type? I tried another three. That is B cell, CD14, and PDC. So the good thing is CellDEEP seems very good. In these four clusters, three of it is a CellDEEP is the best. And for the PDC, it's single cell who is the best. But you probably noticed that the CellDEEP strategy, which outperforms in each cell type, is different with others. You can see in the B cell, oh no, sorry, CD8, this is random DC2, and in that is B cell, it's random mean DC2 is still better. But then in CD14, that is random sum must. So they're different. And also another thing catch my attention is, remember in simulation, pseudobulk is not perfect, but it's very good. So the precision is very high, both in Muscat-Zimmermann, and also the sensitivity is quite higher than single cell. I didn't see it in real data. On the contrary, pseudobulk seems either too conservative or it generates a lot of positive. That confused me. And also, so that's the differences between simulation and real data, right? And also, the strategy performed best in CellDEEP in simulation is not the best strategy here. So I'm thinking, what can cause the issue? We did a lot of tests. I also noticed random seems always better than k-mean, which is also something not I expected. I would expect the method catch the more biological similar cells give the better result, right? No, it's a random get better result. So we did a lot of tests. And then that leads to the part three, which is I'm trying to understand what leads the differences between this result. And basically, one of the reason is the sequence quality of the data. So A and D is for ABC are the UMI plot from the dataset. So basically it's saying how deep it's sequenced, how better the sequencing quality is. For A is the CD8 cells I used in this test. And for B is another CD8 cells I picked from the age-related PBMC data. The good thing is it's also CD8 cells and it's also from human. And you can see higher sequencing quality than the previous one. So previous one is one from 2021. And this age-related CD8 cells is a research from 2025. So with technology development, you can see the sequencing is better. And the last one is the simulation. So you can see simulation has very high sequencing quality. Well, real data is sometimes not that perfect. It's just what it is. Then if we look at this one plot, so in CD8, the low quality one, you can see seedbug have some positive. Single cell have a lot of positive. CellDEEP control it, and they don't have much in common. But in higher quality data, the false positive seedbug start decreasing. Single cell is the same though. And also the false positive cells, it's not really false positive, but if only this method uniquely identify it, it's possible they are false positive. It's just to show, I just want to show how the trend is going now. So they have more thing in common now. And CellDEEP and CellDEEP are detecting lies, um, uniquely identified genes. And then finally in simulation. So you can see simulation is also single cell have a lot of positive, while CellDEEP and CellDEEP control it very nice. And in the middle they have about 20% similar. So it's quite clear that with the sequencing data quality increase, the result actually more like simulation. So I would imagine when our sequencing quality developed, that is technology developed, we would have the same result as simulation, that is CellDEEP. That would lead CellDEEP would be the best. So in the end, key outcomes, CellDEEP is doing what I want. It controls phosphatase and keeps the single cell level information, keeps the synthesis. Random seems better, but there isn't one consistent outperform strategy. Then uh, the differences between simulation and real data and everything, it might come from the sequencing quality. So I'm still working on what affect the differences, like random. My random strategy seems better because I imagine it's coming. I have some result now, I didn't put it here, but it's possible because so when you are doing the k-mean selection, right? You select the cells more like each other. They are not really biologically like each other, but just their read counts, the gene read counts are more similar. So that means you probably also keep the dropout and the zero read counts. So you keep the noise and we try to avoid. That's why k-mean is not as bad. K-mean is worse, but random is better than k-mean. So thank you for your listening.